Hello, everyone. My name is Joseph Dolce. Today, I'm going to be presenting on the gut microbiome and body metrics and providing insights from a study conducted in Bangladesh. I wish I could be sharing my face with you guys today, but every time I record with my face, my computer seems to crash. So this is just going to be how the presentation will go. Let's begin. So how does the gut microbiome influence our body and why should we care? Well, the gut microbiome has an influence on our health and risk for disease. It's important to pay attention to the study of the gut microbiome in order to understand the development of more efficient and effective health interventions being created against diseases. Researchers hope to learn more about the obesity epidemic taking place in countries such as the United States by understanding the correlation between adiposity and the composition of the gut microbiome. This can then allow researchers to potentially lay the foundation for healthcare professionals to offer more personalized treatment against obesity and other diseases as well. So the main question being asked in this research article is what is the possible relationship between the human gut microbiota and anthropometric measurements? What are anthropometric measurements? Well, they're measurements of a person's body size, shape, and composition. So this includes BMI, body mass index, and WC, waist circumference. The goal of this study was achieved through uncovering which genera and families resulted in different measurements, and also looked into what's currently known about the gut microbiome's involvement in diseases, relating it to how differences in diversity and richness in the gut microbiome relates to various diseases such as obesity. So, what is the role of the gut microbiome? Well, the gut microbiome consists of trillions of microorganisms that influence our metabolism, immune function, and energy balance. According to Sabitha Sasidharan Pillai in 2024, an imbalance in gut microbiome composition, known as dysbiosis, has been linked to obesity and type 2 diabetes. Additionally, Pillai mentions how the gut microbiome composition is influenced by genetics, diet, age, and lifestyle factors. And as we grow older as people, our lifestyle changes, different factors in our life change, and as a result, our gut microbiome is going to be changing as well. Pillai also highlights how the microbiota helps digest complex carbohydrates and produce short chain fatty acids. Now, if there's an imbalance in the production of these short chain fatty acids, this has been associated with increased fat storage, insulin resistance, and metabolic disorders, further highlighting how impactful microbiome dysbiosis can be on the entire body. So dysbiosis leading to obesity is something I've kind of hinted at in the past slide. So Pillai in 2024 detailed how obese individuals often exhibit a lower gut microbiome diversity and a higher ratio of different bacterial genera. Dysbiosis contributes to increased gut permeability and inflammation through the release of pro-inflammatory metabolites like lipopolysaccharides. And previous studies have focused on Western and high BMI populations as well, with most research coming from places with larger BMI ranges, meaning there's limited research on gut microbiome diversity in lean populations, and so in places such as Bangladesh. A study done by Heinz Buschardt and Wams in 2018 made it clear that there is a knowledge gap in the function of the gut microbiome that they're hoping to close through research. So while previous studies have established the gut microbiome's role in obesity and metabolism, little is known about its impact on anthropometric markers in lean individuals. So there's a need to investigate whether gut microbiome composition correlates with body measurements beyond BMI, such as waist circumference and waist to hip ratio. And by understanding gut microbiome patterns in lean populations, we can provide insights into how metabolic health can change in various populations beyond obesity-focused research. So how does obesity impact us? I have a graphic here on the right detailing what obese adults may be at a higher risk for. So there's heart disease, high blood pressure, stroke, diabetes, cancer, and osteoarthritis as well. According to Botari and Mantzoros in 2022, the global prevalence of obesity tripled between 1975 and 2016. This led the World Health Organization to describe obesity as reaching pandemic dimensions. Botari and Mantzoros also mentioned that COVID-19 left many individuals quarantined at home and sedentary. So there's believed to be a bit of synergy between COVID-19 and obesity since after the pandemic, there seemed to be a rise in obesity rates as well. So why study in Bangladesh? Well, by examining a leaner population, this research could then be used when addressing metabolic health in diverse communities, including those who are underweight, overweight, or somewhere in the middle. So the title of the paper I'm covering today is The Association Between Gut Microbiome and Anthropometric Measurements in Bangladesh. The main question that we can derive from this title is that can bacteria play a role in body size and fat distribution, even in lean individuals? An important follow-up question to this is which bacteria may be playing an impactful role in anthropometric measurements, as well as investigating what differences there may be between the population in Bangladesh and those living in countries such as the United States. Answering questions like this is how researchers and scientists can move towards a better understanding of the gut microbiome and provides a bit more foundation for understanding how the composition of bacteria in a person's gut may impact their bodily measurements. So some work to address some of the concerns about obesity, which I mentioned in the previous slide. So now we're looking towards analyzing a lean population. On the right here, I have a worker in Bangladesh working in one of the dye industries, just so we can kind of visualize the population we're working with today. So this study was part of a health effects cohort in Bangladesh, and it was investigating gut microbiome and body size in lean individuals. They sampled 400 people and got a final sample size of about 250. To be eligible for the study, you had to be healthy with no major illnesses or antibiotics for at least a month. The body measurements they were looking at went beyond BMI. It was including waist and hip circumference, upper thigh, mid upper arm, things such as that. And in order to extract DNA from these stool samples that they collected, they used a MO Power Soil kit and used 16S rRNA sequencing. They paired this with Illumina as well. Based on what we have learned in lecture, Illumina is a technology that's highly efficient for microbiome studies due to its ability to generate high throughput, accurate, and relatively cost-effective sequencing data. Since Illumina utilizes paired end reads, it has improved coverage and accuracy as well, making it very useful for analyzing microbial communities. The advantages of this technology include its high throughput, which is the highest of any platform to date, but it's also important to note that it has a very short read length. And compared to maybe newer technologies today with a larger read length, such as Oxford Nanopore and PacBio, we're going to keep in mind how this may limit the ability for researchers to assemble genomes found in the gut microbiome. In this study, however, the authors chose to pair Illumina with the sequence Illumina to the 16S rRNA gene amplicons that they sequenced, and this allowed them to identify and characterize the gut microbial composition. So why 16S rRNA? Well, according to Raf Winard in 2019, he knows what we've learned about in lecture about how 16S rRNA serves to identify different bacteria by their genetic differences and pairs especially well when using Illumina sequencing, since it allows for reliable identification of bacterial genera. So when doing data processing, Chime was used. And this is a software used to process sequencing data about microbial communities that were sampled through 16S rRNA amplicons. And so the researchers in the study looked further at diversity analysis, different statistical tests, and also did a false discovery rate, FDR, correction in order to control any false positives. So key terms for this, on the right, I have some abbreviations for the body measurements we're going to be looking at today. And then some other terms on the left you may recognize from class, but I just wanted to explain them for everyone. So we have OTUs, which are groups of organisms that are similar to each other based on DNA sequences. There's also the Shannon Diversity Index, which demonstrates microbiome diversity. There's the alpha diversity as well, which is diversity of bacteria within a person. And then beta diversity looks at diversity among different people, so in between individuals. All right, so the first figure we're going to be taking a look at is table 2 here, and it shows the relationship between gut microbiota diversity and anthropometric measurements. The key takeaway here is that as body size measurements such as mid-upper arm circumference, waist circumference, and waist to hip ratio are all increasing, the overall microbial richness and diversity are decreasing. So where do we see the richness and diversity decreasing? So richness was measured through our observed OTUs in the Chow-1 richness index. So both of these are significantly decreasing as the mid-upper arm circumference, and waist to hip ratio are increasing. So this means that individuals with higher body size measurements tend to have fewer distinct bacterial species in their gut. Next, microbial diversity measured by the Shannon Diversity Index also declines as all anthropometric measurements increase. The Shannon Index accounts for both richness and evenness, so this suggests not only fewer microbial species overall, but that there's also an imbalance in the distribution of these species. So this pattern is overall really important because it allows us to see how lower microbial diversity has been linked to metabolic disorders, such as obesity and other health risks. And these findings suggest that gut microbiota composition may be playing a role in body composition even in a lean population. So moving on to our next figure, this is table 3, and it's detailing how rare bacteria can actually drive metabolic processes. So here we're focusing more on beta diversity and how similar or different microbial compositions are across individuals. The key finding here is that individuals with similar anthropometric measurements tend to have more similar gut microbiota profiles. To analyze this, different distance metrics were used. So first we have the unifrac distances, which measure microbial similarity while considering evolutionary relationships. So there is unweighted unifrac and weighted unifrac. The unweighted is looking at more rare taxa, while weighted unifrac is looking more at common bacteria. The Bray-Curtis dissimilarity, on the other hand, is purely based on microbial abundance and doesn't consider evolutionary relationships. And so when comparing these metrics, we can see that BMI and hip circumference were more strongly associated with gut microbiota differences when using unweighted unifrac. So this is able to suggest that rare bacteria are actually driving these associations rather than common ones, as we can see that when we use the weighted unifrac, there are weaker associations. So overall, we can see that more common bacteria might not differ as much across individuals with varying BMI or HC. Overall, this reinforces the idea that gut microbiota composition varies with body size and composition. It also shows that rare bacterial taxa may play an important role in these differences. So the significance of seeing that there is rare bacteria involved in this is that rare bacteria can actually produce unique metabolites that influence digestion, fat storage, and energy regulation. They may also contribute to metabolic pathways that impact how we absorb nutrients or produce inflammation in our body. Even though if these rare taxa exist in small numbers, they still have a profound impact on these processes, these pathways. These rare bacteria also can be used as therapeutic targets. So they can become targets for probiotics or microbiome-based therapies aimed at improving metabolic health, particularly in populations with distinct body composition patterns. So moving into our next figure, we have table 5 here, which is looking at 4 significant bacterial genera and families, which have a strong association with the anthropometric measurements that we've been looking at in previous figures. So what I really want to highlight here is that as we go down the table, the percentage present increases, but we're also going from genus to family. So this kind of, what I first didn't really understand about microbiome research, I thought, maybe a little not fully understanding it, but I thought it'd be pretty easy to figure out what specific species could be influencing all this, but it's not exactly that simple. So we have to look a bit further out. So rather than species, we look at genus, and rather than genus, you can look at family as well. So when you look at family, you see a bigger, wider picture, you're going to have a greater percentage present, but then it gets a bit harder when figuring out what genus in that family may be having a profound impact. So I think this graph does a great job of showing that and also highlighting 4 taxa that are going to be really important in understanding future research that may come out about maybe specific species that can have an important impact. So what I really want to conclude here is that these findings support that the idea of gut microbiota composition being closely linked to body composition in certain bacterial groups may play a role in metabolic health. The consistent associations across multiple anthropometric measurements highlights the robustness of these relationships, which hopefully we're going to go into a bit more now. Bacterial genera can impact measurements, as we've talked about a little bit. We have a heat map here for our next figure in order to show this. So in the red, or actually, let's start. On the x-axis, we have our different measurements. On the y-axis, we have our different genera here. And in the red, we have positive correlations. So the more red, the more positive correlation of as measurement increase, so does the abundance of the genera. Blue meaning negative correlation, so greater the measurement, lower the abundance of the genera or genus. So what is being demonstrated here? Some key takeaways I really want to highlight is the Oscillospira, which was one of the genera that we just highlighted, the genus, it was a genus we just highlighted. It has a negative correlation with all anthropometric measurements, meaning that their abundance decreases as body size increases. So this suggests it may play a potential protective role in maintaining a leaner body composition, as we can see that blue band there going through the Oscillospira genus. And there's also the Ruminococcus genus, which is another key bacterial genus that was inversely related to MUAC, WC, and WHR. It's a bit lighter blue than Oscillospira, but it's interesting to see how it's also related to that. And we also have some positive correlated ones. We can see Lactococcus has a pretty positive relation for thigh and BMI. I think Enterococcus and Streptococcus, those are pretty common genera that we see a lot in our microbiology classes. So it's interesting to see how, maybe genera we've already learned about can have these impacts that maybe we didn't know before. All right, so looking more at Oscillospira, this is more so highlighting that negative correlation it has with anthropometric measurements. So in these graphs here, they're looking at various anthropometric measurements. Individuals are grouped into tertiles based on Oscillospira abundance along the x-axis, while the y-axis represents the least square means of each body measurement, shown as black squares with error bars. So there's a clear dose response decrease in Oscillospira abundance as anthropometric measurements increase. In the group A above, they're using the Spearman's partial correlation analysis, while on panel B, they're using ANOVA to compare mean differences across groups. And it's interesting to see how different measurements still are showing these sort of negative, inversely related relationships. So both analyses support the trend that Oscillospira abundance declines as measurements such as BMI, waist circumference, and hip circumference are increasing. All right. So last figure I want to go over is actually a supplemental table that I had to go looking for in the paper, and it highlights how sex differences are apparent for Acidomycoccus, a genus we had highlighted before. So this table examines differences in the relationship between Acidomycoccus abundance and anthropometric measurements. The analysis done in this paper found that associations between Acidomycoccus and BMI, WC, and HC were significantly different between men and women. You can see the p-values I've highlighted here for BMI, WC, and HC are all significant. And these positive associations were significant in women, but not in men. So this suggests that there's a potential sex-specific role of Acidomycoccus in body composition. So this highlights the importance of considering sex differences in microbiome research, as microbial influences on metabolism and adiposity may not be uniform across populations. So further studies, I think it's definitely great if they explore the mechanisms potentially behind these differences, whether it's hormonal, genetic, or lifestyle factors, and just sort of observing how these sex-specific relationships can have an impact on our body as well as bodily measurements. All right. So restating my bottom line up front, we've seen today that there are significant associations between gut microbiota diversity and anthropometric measurements such as BMI, waist circumference, mid-upper arm circumference, and waist hip ratio in a rural Bangladesh population. It's also important to note that lower fecal bacterial diversity was consistently linked to higher anthropometric measurements, suggesting a potential role for gut microbiota in adiposity. So, what were some of the specific genera that we're looking at today? Specific genera and families we're looking at today. So, the genus Acilospira had lower abundance as measurements increased. This suggests we talked about its potential protective role in maintaining a leaner body composition. There's also the family S247, which also showed an inverse relationship. This was shown in one of the larger tables I was unable to include, but I wanted to mention this as well here. And it reinforces the pattern that we saw for Acilospira. Acidaminococcus, we saw a positively associated BMI, WC, and HC, meaning individuals with higher body measurements tended to have greater abundance of this bacterium. And we also saw how it may be related to sex-specific microbiota effects as well. And Rhaminococcacea was another key bacterial family that was inversely related to MUAC, WC, and WHR, further linking microbial composition to body metrics. All right, so when it comes to targeting the microbiome, these findings offer a potential pathway for addressing metabolic health and obesity by targeting gut microbiota composition, which could influence body weight and fat distribution. Understanding the role of Acilospira and other microbial taxa in regulating adiposity could lead to microbiome-based interventions in managing obesity and related diseases. These insights are relevant for global health, especially in low-income populations where interventions for metabolic diseases are a bit more limited. This study highlights the importance of microbiome diversity as a health marker and suggests further exploration of gut-derived short-chain fatty acids, such as butyrate, in maintaining metabolic health. So it's important to know some of the limitations of this study. We still don't fully understand the mechanisms behind specific microbiota influencing, or behind which specific microbiota influence anthropometric measurements, particularly in how these interactions translate to health outcomes. It's also important to note some of the sequencing restrictions. I talked a bit earlier about Illumina and how it has short read lengths. So maybe the result of the study could have deferred had the authors used a different sequencing technique that utilizes a larger read length. So it'd be interesting to see what could result from that and what kind of picture they'd be able to piece together. The authors also suggest that further research be done with metagenomic sequencing in order to identify the functional roles of specific species within these genera. So this is going to take a lot of time, but they suggest that longitudinal studies be done in order to assess how changes in the microbiota over time relate to shifts in body composition. So as we work to improve our metabolic health, I just have this to say in summary, that the findings reinforce the idea that gut bacteria are closely tied to body size and composition. We saw that lower bacterial diversity was linked to larger body measurements, and specific bacteria like Acilospira were consistently associated with leanness. On the other hand, Acidomenococcus showed a strong link to higher body measurements, particularly in women. And these results suggest that the gut microbiome plays a significant role in metabolism and obesity. However, we still don't fully understand the mechanisms behind these relationships, and further research should help us uncover whether modifying gut bacteria could be a potential strategy for improving metabolic health. Lastly, these are my discussion questions for you guys. So one is involving potential interventions. So using what was found in the study, could researchers and healthcare professionals use specific bacterial genera linked to leanness or obesity to help patients? So I think about that, what could be done with what we know currently. What is the relationship with global health? So given that the study was conducted in a lean and low income population, how would the results be different if it was done somewhere like the United States or United Kingdom, where there may be a higher income setting where more diets and lifestyles are present? What may be the reasons for sex differences? So we saw that with Acidomenococcus. Could it be biological factors, lifestyle factors that are contributing to this difference? Just any predictions you guys may have, um, and different sequencing choices. So, what would you guys have used? Do you think Illumina was the right choice here? Um, could a different could a different sequencing tool be used instead, and what would the impact be? And what could future research be? So if you were tasked with completing research to build off of this study, how would you design the experiment and what would your main research question be? If you um, if you like, you may even relate it to gaps in our current knowledge and what was left by the authors of this study to further look into. So yeah, these are my references. Thank you guys so much for listening. I hope you have a great day.